Hello, good evening friends. So, in this video, I thought of starting with recent advances, topic flow cytometry. So, I will be covering about what is flow cytometry, what are the different parts in flow cytometry, how it works, what are the unique advantages and its application in microbiology. Starting with what is flow cytometry. It's a fluidic system which involves passing of cells in a single file which will analyze both qualitative and quantitative parameters. So what is different from fluorescent microscopy is in fluorescent microscopy it will only analyze qualitative parameters, whereas in flow cytometry there is additional of quantitative parameters as well. Coming to what are the different parts in flow cytometer. So there is sample injection port where the sample goes in single file. So we will have both stained and unstained cells. So what are stained cells? They are stained with fluorescent antibodies actually if we are looking for particular antigen. So there will be laser source which will hit the cells leading to forward scatter and side scatter. Forward scatter is in the straight line which is detected by photodiode. Forward scatter detection is by photodiode and side scatter is around 90 degree which is detected by photomultiplier tubes. And there is also emission of fluorescence which is detected by fluorescence detectors. From the laser source, when it hits the cell, in the forward scatter, the range of 5 to 10 degree of forward scatter is detected by photodiode. It indicates the size of the cell, and larger the cell, more is the forward scatter. Around 90 degree, we have side scatter which indicates the inner details of the cell structure like mitochondria, nucleic acid, etc. which is detected by photomultiplier tubes. What are photomultiplier tubes? There will be conversion of photon into electrons using photoelectric effect, and from that there will be generation of voltage graphs. So as the laser beam passes through the cell, laser beam is behind the cell now, into the cell and in front of the cell. That time we will have voltage graph. The peak is when the laser beam hits the cell. So the graph will give quantification like height of the voltage, the width of the voltage and the area covered. So among the cells, the stained cells which will emit fluorescence is also detected, as already I had mentioned. But other parts during fluorescent detection, we also need diachronic mirror and filters, some of the filters. So what is diachronic mirror? It is a beam splitter which reflects beam of certain wavelengths, and filters. There is band pass filter, there is long pass filter and short pass filter. Band pass filters which will allow range of wavelengths to pass through. Like if it is named BP 730 bar 30, it means that it will allow 730 nanometer plus or minus 30 nanometer. Long pass filter, that will allow wavelengths longer than the specifications mentioned. In short pass filter, it will allow wavelengths shorter than the specifications mentioned. To sum up what happens in flow cytometry, first there is a laser source which will emit the laser beam passing through the focusing lens, passes through the cell. There is forward scatter, there is side scatter which I have already explained, and there is fluorescence collecting lenses. Then it will pass through diachronic mirror. As I already explained about diachronic mirror, it will reflect the beam of certain wavelength. Then it can pass through band pass filter, then some of the wavelengths can pass through long pass filter, and we can have multiple fluorescence detectors based on certain wavelengths that we need. Coming to the workflow, so first there is passage of stained and unstained cells from the sample injection port, then subjected to laser beam leading to forward scatter, side scatter and fluorescence detection. It is connected to digital system where analysis is reflected in the form of dot plots, histograms, counter plots and voltage plots. It has its application in microbiology in variety of fields like identification part in bacteriology, mycology, virology, and in immunology, in virology, separately to analyze the quantity of the infected cells by the virus, then pathogenesis, viral host cell interaction, etc. In environmental surveillance, in epidemiological and research purposes, and in food industry, which I will explain in detail each one. It is helpful in identification of various bacterial counts like viable, dormant or dead cells, and early detection of mycobacterium species. Intracellular pathogens like chlamydia and rickettsia. In mycology, it has been found to identify various species and strains of candida, especially helpful in recurrent candidal infections. In virology, when it is used, it is also called as flow virometry. It will give the number of viral infected cells. It will also help in understanding of viral host cell interaction, which will give details of viral entry, viral replication. Helpful in identification of virus by using fluorescent antibodies targeting the known surface antigens. So in few of the studies, it was found that it was also helpful in antiviral susceptibility detection. Coming to intracellular parasite detection, research has been done on identification of plasmodium and proved to be helpful. And also research is being done on anti-parasitic drug susceptibility. So antiviral drug susceptibility, anti-parasitic drug susceptibilities, which are not routinely done, is also found to be helpful from flow cytometry. In environmental surveillance, it is especially used in identification of any contamination and in root cause analysis of water contamination, the source identification, the water purification system analysis. All this it has been used. In food industry system, where in milk and milk products bacterial counts are detected, even in probiotics, etc., culture is being used more commonly. Other methods that are possibly used are also PCR, ELISA, microarray are also present. But flow cytometry is superior because they are able to detect viable but non-cultivable bacteria also. So what are viable non-cultivable bacteria? Viable, that is the metabolic activity in those bacteria are present, but they are not actively dividing cells. Those are also identified by flow cytometry. Coming to limitations in flow cytometry, which is same as in other methods also, like the quality of sample collection or transport. If that is poor, then the analysis part will not give the accurate results. Now, another important limitation is if there is cellular aggregation, as in formation of biofilm, etc., the digital analysis may give false results. There may be interference in the digital analysis. So coming to sepsis and immunology part, research has been done in studying host cell and pathogen interaction part. And also in immunology, already we know that it is used in CD4 counts, CD8 counts detection, NK cell count detection, and in cancer immunology using monoclonal antibodies, it is very much used in the cancer diagnostics. So, to finally summarize flow cytometry, it is a fluidic system which will analyze cell count, cell size and details in the cell using forward scatter detection, side scatter detection, fluorescence detection, and its uniqueness is in its ability to quantify, its ability to identify viable, non-cultivable cell, in also understanding the pathogenesis, host cell pathogen interaction, in also detecting the intracellular pathogen, which is very difficult with the usual conventional methods. So, please like, share and subscribe. We'll meet in the next video.